Well, hello again, everybody. Welcome back from the break. I think I've sorted out the issues with Zoom. We'll see what happens. Let's see if we can't get the remaining sections of this chapter recorded and ready for you to use. So when we finished off at the end of the last section, I presented this concept check question. So let's take a quick look at it again. You find a fossilized skull that has a C14 to C12 ratio of 1/16 that of modern day skulls. Approximately how many years old is the fossilized skull? Well, of course, the first thing we need to figure out is how many half-lives have passed on the basis of 1/16 of the C14 remaining. And if you recall, a half-life is defined as the time required for half of the isotope to decay. So after one half-life, you have half of what you started with. After two half-lives, you have a quarter of what you started with. After three half-lives, you have an eighth of what you started with. And after four half-lives, you have 1/16. So immediately we know that 4 half-lives have passed. So let's make a note of that, 4 half-lives. Then we know that the half-life of C14 is approximately equal to 5,700 years. So it's going to be 4 times 5,700, which equals 22,800 years old, approximately. So it's a pretty straightforward calculation. Of course, you should be familiar with how to do that because we did these sorts of calculations in the paleontology lab, and hopefully you mastered the calculations there. So let's move on and take a look at the next section. So in this section, we're going to be touching on some of the major points in the history of the evolution of life. And we're going to encounter a couple of ideas that hopefully you're familiar with, or maybe you're not so familiar with, but are key ideas that you need to be familiar with and to understand the significance of some of these ideas. So the first thing that we recognize from the fossil record is that the earliest history of life, and for the longest time period during the history of life, life was present as purely prokaryotic cells. So bacteria and archaea represent, for the most part, the predominant life forms that have been present on the Earth, and certainly for a significant time period from the point at which life began, roughly 4.2 to 4 billion years ago, until around roughly 1.5 billion years ago, based on molecular clock evidence, life was strictly prokaryotic. Now, that doesn't mean there wasn't a lot of things going on just because you had a single type of cell. One of the things that we're going to encounter, and one of the most important ideas that we need to take on board, is that life does not just adapt to changes in the environment. At certain times in the history of life, we know very well that life itself has changed the environment, or at least some small subset of life has changed the environment, which has forced all other life forms, and perhaps those life forms themselves, to adapt to those changes. And that is very much the first thing that we need to take into account. So when we think in terms of the oldest fossils, and I've already mentioned this in a previous section, the oldest fossil that we have that has been absolutely dated, so using radioisotopes, particularly the uranium decay series and perhaps potassium argon, but most notably the uranium decay series, the oldest fossil on record is 3.48 billion years old. It originates from a rock formation in northwest Western Australia. And I've had the rare opportunity to actually visit the location where that oldest fossil was taken from. And I can tell you there's not really a whole lot to look at of great interest until you recognize what it is that you're actually looking at. And what you're looking at is a remnant of a reef-like structure built by cyanobacteria. These are fossilized stromatolites. And that means we're looking at bacteria that have the capability to carry out photosynthesis, and specifically oxygenic photosynthesis. And that's the key to this first major step in the history of life. So here you can see some examples of stromatolites. We've already seen some photographs of these in a previous section. Again, we reproduced this picture of the shark bay stromatolites, and I've visited those as well. And so I can assure you that that's exactly what they look like. In the area where I visited, it's actually a national park, so you can't actually wade out to get in amongst the formations, but I stayed at a nearby sheep station that has been turned into a tourist accommodation as well. And the people who ran it said that if I'd been there for a few days longer, because I was only there for 2 nights and a day, there would have been an opportunity to go a little bit further around the bay outside of the national park, where I could have actually walked out amongst the formations. They would have been perfectly happy to take me there. So I can tell you that these formations look exactly like this, and these are cyanobacteria that have been growing in this location for approximately 3,000 to 4,000 years. And they are essentially identical to the fossilized material that has been dated back to 3.48 billion years ago. So we know what we're looking at here, and we know from the fossil record that this is what we're looking at. So one of the things that is important to understand here is that these are photosynthetic, and that has consequences, and specifically oxygenic photosynthesis. And that means they're generating oxygen. Why is this significant? Well, if you recall from the previous section, the early atmosphere of the Earth was a reducing atmosphere. There was essentially no oxygen present. So here we have a major ecological change being caused by the presence of certain life forms, the introduction of oxygen into the atmosphere. And we know approximately when this began to have some significance. So the oldest fossils are these cyanobacteria, 3.48 billion years old. Where the next evidence from the fossil record starts to take on significance are the appearance of these reddish bands that we find in many rock formations dating back to somewhere around 2.5 to 3 billion years ago, because these are rusted iron oxide. And what that tells us is that there is now sufficient oxygen present to start reacting with the iron dissolved in the oceans, the primordial oceans at this point. Because if there was a reducing atmosphere and the Earth was basically functioning by reducing chemistry, then reduced iron is very, very soluble in water. And so the oceans at this time would have contained a lot of soluble iron and would have looked very differently from what we would have, or what we would expect today. They wouldn't be the bluish color that we see today. They would have been a dark brown, ugly greenish color. And that would have been because of the presence of all of this dissolved reduced iron. But as the oxygen levels start to increase due to the activity of the cyanobacteria, well, we know that iron is very, very reactive with oxygen, and it rusts. They combine together very easily. So as the oxygen starts to get introduced into the water by the cyanobacteria, the oxygen starts to react with the iron. We get iron oxides. And the thing about iron oxide is it is very, very insoluble. So the iron is now changing from a soluble form to an insoluble form. And that means it starts to precipitate out. And that's the origin of these reddish bands. And we know this begins somewhere around 2.5 to 2 billion years ago. That's when we find the initial appearance of these bands. And as the oxygen levels continue to increase, more and more of this iron precipitates out. And many of the large deposits of iron ore, which is essentially rusted iron, that we mine to get access to the iron to make iron and steel, is the direct product of this so-called oxygen revolution, the introduction of oxygen into the environment by the cyanobacteria. And quite ironically, or maybe parenthetically, some of the largest deposits of rusted iron exist in the Pilbara region of northwest Western Australia, adjacent to this region where we have the stromatolites growing in the ocean, but also evidence of very, very old cyanobacteria in the form of stromatolites in the fossil record. Maybe not so coincidentally as you might imagine. But the significance of this is that while we see the increase of oxygen and the precipitation of the iron, very little of that oxygen at this point is available outside of the water because it's going to combine with iron. And in fact, oxygen levels in the oceans would not start to increase significantly until all the iron had reacted with oxygen and precipitated to the floor of the ocean. Then with nothing more to combine to, the oxygen levels in water would start to increase, and the oceans would take on a more oxygen-rich nature. But oxygen does not dissolve particularly readily in water. So once the oceans become saturated with oxygen, which would happen relatively quickly, because again, oxygen is not particularly soluble in water, then the oxygen starts to percolate into the atmosphere and we start to see a rise in atmospheric oxygen. Now, today we experience oxygen levels in the atmosphere of around 20%. That's our measured concentration of oxygen in the atmosphere. The sort of levels that are generated at this point are substantially lower than that. Probably only in the range of 2 to 3, maybe 4% oxygen in the atmosphere. But that is still significant because up until this point, life has been anaerobic, functioning in the absence of oxygen. And based on our experience with anaerobic organisms today, the vast majority of anaerobic organisms are killed by the presence of oxygen. Oxygen is extremely reactive. You may recall from Bio 1 that oxygen is extremely electronegative, second most electronegative element, and that makes it extremely chemically reactive. And as a consequence, if you don't have a mechanism in place to neutralize the oxygen, and electron transport chain is the perfect tool, then you're going to suffer from the effects of oxygen, the chemical reactivity of it, and likely you're going to die. And so we would expect, although there's no evidence in the fossil record because there's a limited amount of fossil record and most organisms do not fossilize that would have been present at that time. We're very lucky, in fact, to have the cyanobacterial fossils. But it is very likely that a significant mass extinction event took place at this point in time. We don't have any direct evidence for that, but logic dictates that if life is largely anaerobic at this point, and you get the introduction of large amounts of oxygen via photosynthesis, that is not going to be good. And so the vast majority of organisms probably died out at this point, simply because they could not cope with the oxygen. Some of them took a second route to survival, and that is they moved to those areas that remained largely anaerobic and are probably in some way related to the number of anaerobic organisms that we still have present today. So you may recall that there are anaerobic bacteria still present in the environment. Some of them are obligate anaerobes, meaning they have no choice in the matter and therefore have to survive or live in an oxygen-free environment. And they are probably somewhat descendants, and we can't say directly because we have no real evidence, but we know that organisms did survive by moving into anaerobic environments. A very small number that were lucky enough to have a pre-existing mutation had the ability to at least survive the presence of oxygen, probably because they already had some, if not all, of a electron transport chain or some mechanism that allowed them to neutralize the oxygen in some fashion. And that would have allowed them to at least survive the presence of oxygen. by neutralizing the oxygen, they did not have the problem of oxygen's reactivity, and so they could survive the presence of oxygen. At some point along the way, we don't know exactly where, but we can probably hypothesize as to how it took place, as you'll learn when we talk about bacteria in an upcoming chapter. Bacteria, and archaea for that matter, are very profligate or promiscuous, if you want to use a different term, in terms of their DNA. They do exchange DNA quite readily, either by taking it up directly from the environment, or by directly exchanging it from cell to cell, or by using viruses that specialize in attacking bacteria, called bacteriophages, that can package bacterial DNA rather than their own DNA and transfer that to the next cell that they infect. And so there is the ability amongst prokaryotes, and particularly bacteria, to exchange genetic material. And if you've got organisms that contain bits and pieces of what today we call cell respiration, and they're exchanging DNA, you might expect that different parts of cell respiration found in different cells would at some point over time appear in a single cell. And you have the origin of cell respiration. And of course, that changes life completely, because now you have access to large amounts of energy in the form of the ATP produced through cell respiration. It would not have happened overnight. It probably took a long period of time for these pieces to come together. But we know that some of the components of cell respiration continue as separate parts, or separate parts of photosynthesis for that matter, continue as separate parts within bacteria, because there are bacteria that have a subset of the components of both cell respiration and photosynthesis. And so it is not that difficult to hypothesize the origin of cell respiration and photosynthesis by the exchange of DNA between bacterial cells. And the evidence for that is pretty substantial. And the ultimate evidence for that is this increase in oxygen at this point in time. So we have both organisms or life making a substantial ecological change to the environment, the production of oxygen, but then that forces the remainder of life forms, and perhaps even these life forms themselves, to adapt to the presence of oxygen. This is the oxygen revolution, and it is a key change in the trajectory of the evolution of life, because now you have the opportunity through cell respiration, taking advantage of the reactivity of oxygen, to start generating large amounts of chemical energy in the form of ATP to become or to change the direction in which life would evolve, because you have the energy to support that evolution, I guess. We'll talk more about that when we talk a little bit about the Cambrian explosion a little bit later in this presentation. In fact, a little bit later in this section. So that's the first major change that we associate with the in the timeline of life's evolution. Now, one of the things that then highlights the next part, or the next important step, is the appearance somewhere around, we think, 1.5 billion years ago, based on molecular clock evidence. There is some fossil evidence that has been interpreted as eukaryotic cells, but we're not entirely sure that that's the case. This is the way some people are interpreting them, but molecular clock evidence suggests that eukaryotic cells originated roughly 1.5 billion years ago. Now, the origin of eukaryotic cells has been the subject of a lot of controversy, because there are some parts of eukaryotic cells that are really easy to explain, but there are other parts of eukaryotic cells that people have struggled with trying to explain, and there are some parts that people are still struggling with. So let me show you this timeline, because it gives you some clues. So again, casting your mind back to what you learned in Bio 1, you may recall that all eukaryotic cells have what is called the endomembrane system. And that consists of the nucleus, the endoplasmic reticulum, the Golgi apparatus, the transport vesicles that link them together, the cell membrane, lysosomes, the ribosomes. They're all considered to be part of the endomembrane system. And it is not difficult to imagine the origin of the endomembrane system through invagination of the plasma membrane. These are membranes that, the membranes of all of these organelles resemble very closely the plasma membrane. And so it's not difficult to imagine the origin of the endomembrane system, and certainly the membrane-bound components of it, as being the product of invaginations of the plasma membrane. We already see that the origin of internal membranes in prokaryotic cells, which you may recall by definition do not contain membrane-bound internal compartments, but they do contain, in the case of aerobic cells and cyanobacteria, internal membrane systems that are derived from invaginations of the plasma membrane of those cells. And in fact, those membrane systems remain connected to the plasma membrane, indicating very clearly that they are the product of invagination. So if in modern prokaryotic cells we see those sorts of invaginations giving rise to internal membranes, then it's not difficult to imagine the origin of the internal membranes of a eukaryotic cell in much the same way. Where people have struggled for a significant amount of time is trying to explain the origin of bacteria, of mitochondria and chloroplasts. And an individual that is actually shown in an earlier photograph, so let me go back. This researcher, Lynn Margulies, was the first to propose, based on her observations of bacterial life forms, that the origin of mitochondria and chloroplasts could be best explained as considering them to be symbionts that were originally free-swimming bacteria, but were in some fashion captured by early eukaryotic cells, and rather than being turned into a food item, were retained within the body of the eukaryotic cell. And she based this on the fact that chloroplasts and mitochondria show some bacterial characteristics. Now, when she proposed this, and I believe it was around 30 years ago now, 30, 35 years ago, most people said, no. There's no way that these could be derived from bacteria. And in fact, every time she gave a talk at a conference proposing this idea, she was roundly criticized and told that she was wrong. But rather than give up, she persevered, and she continued to do research looking at the organelles, and other people's work continued to look at the organelles and compare them to bacteria. And over time, evidence started to accumulate that further reinforced Margulies' observation that these are probably endosymbionts. And so after about 20 years of struggle, finally, enough evidence accumulated that people had to say that, in fact, Margulies was correct, that bacteria, and specifically aerobic bacteria and cyanobacteria, respectively, represent the origin of mitochondria and chloroplasts. So what is the evidence that supports this? Well, the first piece of evidence that supports this is the simple observation that both of these organelles reproduce separately from their host cells. And they reproduce in a fashion that is identical to bacteria, that is binary fission. So rather than replicating when a cell replicates, they only replicate after the host cell has replicated, and then they undergo their own replication process. The second piece of evidence is that they contain ribosomes that are identical to bacterial ribosomes. Now, there is a distinct difference between bacterial ribosomes and eukaryotic ribosomes. Eukaryotic ribosomes, like bacterial ribosomes, are made up of two components, but both of the components in eukaryotic ribosomes are slightly larger as measured by something called Svedberg units, which is basically a measure of how these particles are precipitated by a centrifuge, the rate at which they precipitate by the centrifuge and where they turn up in a density gradient. And so the measured sizes of a eukaryotic ribosome is a bit larger than the measured size of a bacterial ribosome. But what really drives this home is the fact that bacterial ribosomes are sensitive to two antibiotics that shut them down and prevent protein synthesis. And the ribosomes in both chloroplasts and mitochondria are also sensitive to exactly the same antibiotics. These antibiotics, however, have no effect on eukaryotic ribosomes. Then you also have the fact that both mitochondria and chloroplasts contain their own DNA, and it is in the form of small circular DNA molecules, very reminiscent of the and now I've had a brain conniption and I've forgotten the term. Plasmid. So the small circular DNA, auxiliary DNA molecules that are found in bacteria called plasmids, and are in fact the DNA that is readily exchanged between bacterial cells. Is found, molecules similar to that, small circular DNA molecules, are found both in bacteria and in chloroplasts. And that that DNA contains genes that are involved in cell respiration and photosynthesis, respectively. And the ribosomes of the I keep wanting to say bacteria, of the mitochondria and the ribosomes are there to produce the proteins that are encoded in those genes. Now, as a result of the endosymbiosis, some genes from the ancestral bacteria of the mitochondria and the chloroplast have been transferred to the nucleus, a process known as horizontal gene transfer. So we no longer consider these two organelles to be independent or autonomous. The term we use is semi-autonomous, meaning they contain some of their own genes, but they are dependent on genes that are now incorporated into the nuclear DNA. So there is a body of evidence, and that's not all of it, but those are the main points that support Margulies' original contention that mitochondria and chloroplasts originate as free-swimming bacteria that were captured and turned into endosymbionts by early eukaryotic cells. The timeline, or the order in which the capture took place, almost certainly was mitochondria first and chloroplast second. And the evidence for that is very simple. If you look at eukaryotic cells, universally, eukaryotic cells either have mitochondria or have remnants of mitochondria. Now, those remnants may be in the form of a organelle that is derived from the mitochondria but no longer has active mitochondrial function. But the evidence that tells you that they did have functional mitochondria at one point is the fact that those mitochondrial sequences that are found in the nucleus of eukaryotic cells are present in these cells that lack functional mitochondria. And that tells you that at one point they did have mitochondria. Those genes could only have come from one place, mitochondria. And so the fact that there are no longer functional mitochondria in these cells is not evidence that they never had mitochondria. The fact that the genes are present is an indicator that they did have functional mitochondria. And when we talk about eukaryotic cells in chapter 28, or the protists in chapter 28, I'll give you a bit more information about the significance of that. And then only a small subset of eukaryotic cells contain plastids or chloroplasts and other organelles that are closely related to chloroplasts. The two that you may have come across are chromoplasts, which are found in the petals of flowers, and leucoplasts, which are found in cells that specialize in storing starch. So you'll find those in potatoes and other plant structures that specialize in storing starch. That's what a leucoplast is used for. And so the fact that only a small subset of eukaryotic cells, most notably algae and plants, contain chloroplasts clearly indicates that chloroplasts were the second of the two organelles to be generated as a consequence of the endosymbiosis process. And we are so confident in this idea that it has now been encapsulated in a theory. So it's called the endosymbiotic theory of the origin of mitochondria and chloroplasts. And again, if you recall from Bio 1 or from your science education in general, a theory is something that has such support that we consider it to be as close as we are ever going to get to absolute truth. And while we may modify the content of the theory, the basic premise at the heart of the theory, in this case, that the mitochondria and the chloroplast are derived from ancestral free-swimming bacteria, does not change, even though we may refine the theory a little bit more with the as we get better and newer data. We still subscribe to the original premise. So we may modify the theory, but we don't throw it away with new data, because if that new data supports the theory, which in this case, the new data has done so. So that's the next major step. This is the origin of eukaryotic cells. And you can see why this might be very important. Where are we still struggling to try and understand the origin of eukaryotic cells? Well, the major piece is the cytoskeleton. Where did that come from? And also attached to that, the origin of cilia and flagella in eukaryotic cells, because they are very, very different from the flagella that are found on prokaryotic cells. Whereas prokaryotic flagella are separate organelles that are generated from their own suite of genes, flagella and cilia in eukaryotic cells are extensions of the cytoskeleton and literally extensions of the cytoplasm. They're completely surrounded by the plasma membrane. Hopefully you learnt that back in Bio 1. And so to try and understand where the cytoskeleton came from is something that is distinctly different from the bacterial flagellum has been problematic. We do think that the genes for at least some of the components for the cytoskeleton have precursor analogs in bacterial genomes. There is some evidence to support that, which may give us some clues as to the origin of the cytoskeleton, and it's probably not terribly surprising. But we haven't been able to come up with even a supportable hypothesis as yet as to exactly what is the origin of cilia and flagella and the cytoskeleton as the progenitor of cilia and flagella. So that still remains to be solved, but at least we can now propose a basic origin for eukaryotic cells. So that's the next major step in the history of life, and that's another high point that you need to be familiar with. So once we have the early eukaryotic cells, the next thing is multicellularity. And that almost certainly arose a number of times independently amongst the ancestors of the three major eukaryotic kingdoms, meaning fungi, Plantae, and Animalia. So multicellularity amongst animals, multicellularity amongst plants, multicellularity amongst fungi are probably independent events, because all of those kingdoms can be traced back to unicellular eukaryotic protist ancestors, or at least we have hypotheses that trace them back to unicellular protist ancestors. And in the case of fungi, there are in fact unicellular fungi. And when we do chapter 31, which is the fungus chapter later in the semester, we'll talk about the origin of kingdom fungi, and we'll talk about the unicellular fungi there. So the onset of multicellularity is another major step. We can't pinpoint it exactly, but we do know that multicellular forms start to show up in the fossil record somewhere around 1 to 1.5 billion years ago. That's when we start to find evidence of multicellularity, both through molecular clocks and also in some very early fossil records dating to around 1 to 1.2 billion years ago, as the note points out. Perhaps the clearest evidence, however, of multicellularity is in the form of these fossils. These are being interpreted as early stages of animal embryonic development. Certainly the features that we see in these fossils are very characteristic of early embryonic development. When we do chapter 47, again, a little bit later in the semester, you'll be able to put these side by side with what you learn about embryological development. You'll recognize why these are considered to be animal embryos. These date to about 570 million years ago. So if you're thinking in terms of the timeline, this predates the Cambrian explosion. So the Cambrian period begins around 525 million years ago, if I remember rightly. And these date to 570 million years ago. So this puts them back squarely in the middle of the Ediacaran. And of course, we've already seen previously that we have evidence of the origin of the animal, of some members of the animal kingdom back at that time frame, and perhaps even earlier than that, based on molecular clock evidence, particularly for the basal groups of the animal kingdom, which are the cnidarians and the porifera, the jellyfish and their relatives, that's the cnidarians, and the porifera, which are the sponges. They may actually have their origin even earlier than the Ediacaran based on molecular clock evidence. And they are all multicellular, and they have embryological stages that would perhaps look a little bit like this. But these are coming from the middle of the Ediacaran period. So we know that around this time period, we obviously have multicellular organisms, both from the fossil evidence from the Ediacaran and also from these fossils. So multicellularity has its origins somewhere earlier than this, as I said, somewhere around 1 to 1.5 billion years ago, based on molecular clock data and on fossil evidence. So initially, multicellularity was probably in the form of colonies. And certainly when we look at the origin of animals, and when we discuss the origin of animals in chapter 32, we'll look at the protists that we think represent the protist origin of the animal kingdom, and they are certainly colonial. And then when we look at members of the algae, we see multicellular algae, not just unicellular, but we see colonial and we see multicellular. So amongst the algae today, we see evidence of all three eukaryotic forms, I guess. So you have a unicellular form, you have a colonial form, and you have a multicellular form. So there you have evidence for a potential pathway from individual eukaryotic cells to cooperative colonies to a multicellular organism with some level of specialization. And almost certainly this is the direction that you see in terms of further evolution amongst the three big eukaryotic kingdoms. Then we have the Cambrian explosion, or what has been referred to as the Cambrian explosion. This is the next major step in the evolution of life, if we're considering animals at least. And of course, given that we are animals ourselves, this is where we place most of our emphasis when we think in terms of the evolution of life. We think in terms of the evolution of animal life. Is that valid? I'll leave that up to you to think about. But remember, life is more diverse than just animals. And so one of the reasons why we do this class is to try and introduce you to that diversity. And hopefully that will be something that we'll be able to do over the course of this semester. Anyway, Cambrian explosion is a period of about 20 million years, that begins with the start of the Cambrian and extends through that first 20 million years of the Cambrian. And the reason why this is important is because within the fossil record, we see evidence of the increasing diversity of animals. Now, this doesn't mean that there weren't already animals present prior to the Cambrian. As you've seen from the fossil lab, as I've talked about here in the previous sections, and even in this section, some of the members of the animal kingdom have a fairly ancient origin, predating even the Ediacaran, where we see fossils that really represent what we think is the first evidence of animals. And based on molecular clock evidence, and this phylogenetic tree is based on molecular clock evidence, even members of many of the phyla that really only become apparent in the Cambrian during this 20 million year period, then the molecular clock evidence for those phyla suggests that they may even have a deeper origin than the Cambrian. That in fact, early members of these phyla may have been present in the Ediacaran. All you need to do is look at the branch points. So here you have the earliest evidence for the origin of sponges and cnidarians based on molecular clock evidence. So this is where we would find the common ancestor of the animal kingdom. If we were looking for it, that's what that branch point represents. Notice it's back here prior to the Ediacaran. So molecular clock evidence suggests that the origin of the animal kingdom lies back in this region. Then we have the cnidarians themselves branching out during the Ediacaran, and you've seen the fossil evidence that supports the origin of cnidarians in their fossilized jellyfish that we saw in the fossil lab, and the other examples that you've seen. And then we have another branch point. We think that the deuterostomes branch out from what used to be called protostomes, which is this other branch, also during the Ediacaran, again, based on molecular clock evidence. This is not based on fossil evidence, because there's very little fossil evidence to support this. This is based on molecular clock evidence. And then we have origin of the two segmented branches at this point. Now, I will admit that saying that the two segmented branches originated or major segmented clades or major segmented phyla of animals at this point from a common ancestor. That's something that later in the semester I'm going to disagree with. And that's based on evidence. And in fact, I suspect that there is a more recent diagram than this, which I probably should include into this presentation. This is an older diagram from an older edition. I probably should update the diagram here. But we don't think that there is a common ancestor for segmentation. And we'll talk more about that when we get to chapter 32. And then you have origin of other organisms at later points in time. And you notice here we've got a tripartite branch point. You may remember when we talked about phylogenetics and the phyloCode that we don't actually allow for a tripartite branch point. We want only 2 branches coming from each branch point. And what this is indicating is that the origin of these 3 is still somewhat open to question exactly which one came first. And until we have sufficient data to identify which of these 3 clades branched out first from this branch point, we allow for a tripartite branch point only until such times if we have better evidence. But notice all of these are originating back here in the Ediacaran. So why make a big point of that? Because there are a lot of people today who feel that using the term explosion is inappropriate, that in fact explosion should be put in quotation marks. So it should be explosion, because it's not an explosive, or maybe not as explosive, an irradiation or diversification of animals as we once thought. It may not be that a whole lot of phyla are originating during the Cambrian explosion, that many of the phyla were already pre-existing, although there were only a small number of representatives of each of those phyla prior to the Cambrian. What is being proposed is that the Cambrian represents not so much a diversification of phyla within the animal kingdom. So the animal kingdom diversifies into the 35 or 36 phyla that we recognize today, but more that while some phyla do originate during this time period, some of the phyla were pre-existing, and some of the major ones probably were pre-existing based on the molecular clock data. And what we're really seeing during the Cambrian explosion is not a diversification of individual phyla within the animal kingdom, but a diversification of the animals within each phyla. So the phyla themselves pre-exist, but what you're seeing now is an increase in the number of animals within each phyla. That's where the diversification is taking place. Not necessarily a rapid increase in the number of phyla, but a rapid increase in the number of animals within each phylum. And there have been 3 hypotheses proposed to explain why that took place. One is geologic, meaning an increase in the level of oxygen. And if you have an increase in the level of oxygen, that means cell respiration now has the ability to create a whole lot more ATP. You have access to a whole lot of extra energy to do things or to create things. One is a ecological hypothesis, and that is a reference to an increase in predation and changes in predator-prey relationships. And that is evidenced by the animals that we see appearing in the 3 fossil beds that represent this Cambrian explosion period. We see an increase in hard parts. We see an increase in spikes. We see an increase in shells, or we see the appearance of spikes and shells and other protective mechanisms. And that suggests that there are some active predators out there. And why would a prey animal, or why would an animal invest resources and energy in producing protective structures if they weren't required. And as we know in evolution, it's all about cost benefit. If it costs you more than the benefits you get from it, you discard it. So if you're investing resources into protective structures, you must be reaping a major benefit from it. And the benefit obviously is protecting yourself from being eaten. So that suggests very strongly this change in predator-prey relationship and an increase in predation in general. Then there is a genetic explanation, because we start to see the development of master control genes in certain groups of animals. And we'll talk a little bit more about those master control genes. We refer to them as Hox genes a little bit later in this presentation. But the point here is that we have an increase in the number of genes that are responsible for guiding the development of animal body plans. And under those circumstances, it gives you now the flexibility genetically to create a broader diversity of body plans, which may include the protective parts, almost certainly would include the protective parts. Now, when you stop and think about these three hypotheses, a change in gene expression or an increase in the number of control genes that guide the development of body plans, which gives you the opportunity to create a diversity of body plans, and particularly protective body plans, but also may allow you to generate structures that allow you to be a better predator, in parallel with the appearance of those body plans and the increase in oxygen that provides access to energy for doing all of this and becoming more aggressive and more mobile. You start to realize that these three hypotheses are not mutually exclusive. In fact, they are mutually supportive. And so this may be an event that takes place because there is the coming together at this point in time of these three different parameters, or changes in these three different parameters: an increase in oxygen levels, an increase in genetic flexibility, and a change in behavior as a consequence of all of these, that is actually the driving force behind this diversification and rapid diversification in the animal kingdom over a relatively short period of time geologically. But we'll get into some more of that later. So the next major step, and the final step that we want to touch on here, is the colonization of dry land. And this occurs in a very specific sequence. So the first colonizers of dry land are plants, but they do so in concert with fungi. And the reason why is because the early plants to colonize land, based on what we see in the plant kingdom today, probably were very poorly adapted to extract nutrients out of the soil and water out of the soil. They may have been able to extract carbon dioxide from the air, but they were not particularly well equipped to extract water and minerals from the soil. But fungi are particularly good at doing this. Because of the body plan of a fungus, it can penetrate into large volumes of soil and extract nutrients and water in large amounts, because their body plan is adapted very well for absorption. And so for plants to colonize dry ground or dry land, they would benefit from a relationship with fungi that would substitute for the missing root system that the early plants and early plant colonizers of dry ground lacked. They would make a really good substitute. And so the earliest evidence of plants colonizing dry land incorporates the presence of fungi, we see that relationship almost as soon as we start to see significant fossils of plants, we start to see significant fossils of fungi associated with them. And so plants and fungi colonize dry land together in partnership, and that partnership continues to this day. As I've mentioned earlier, about 95% of the modern plant kingdom has some form of symbiotic relationship with a fungus. And both benefit from the relationship, or at least the plants benefit from the relationship. And in many of these symbiotic relationships, so do the fungi. And we'll talk more about that relationship when we talk about the fungi themselves. That's chapter 31. Then that provides the opportunity now for animals to colonize dry land. Up to this point, any animals that may have ventured onto dry land could not stay there because there would be no food source for them. And so while we see evidence of animals in the form of early arthropods, similar to millipedes and centipedes, and perhaps even ancestors to scorpions or arachnids, may have ventured onto dry land temporarily, almost certainly they return to water without staying on land because there'd be nothing for them to eat. And so while they may have been able to survive out of water temporarily, they would have to return to water. But once the plants and fungi colonize dry land, then there's now an opportunity for animals in the form of arthropods to start colonizing land because there are habitats and food sources for them to take advantage of. And so the early evidence of animals on dry land is in the form of arthropods, and most notably in the form of insects. Then eventually the ancestors of amphibians would start to move on to dry land because they have adaptations in the form of primitive limbs that had evolved from fish fins, and we'll talk more about that in chapters 32 and 33, and to some degree in 34 as well, that allowed them to support their body weight out of water. It would have been a little bit of a struggle early on, but as limbs became better adapted to dry land, that would allow them to become more mobile. These would be early amphibians, and they would have food resources to utilize because there were the plants and the insects already present. So they would have food resources that they could utilize. Of course, they would have one drawback: returning to water for reproduction. But eventually a new group of terrestrial tetrapods would evolve that didn't even have to do that. These would be the amniotes, either in the form of amniotes that lay eggs with shells, or amniotes that retain their eggs in the body of the female and support the development of the embryo via a placenta. So this would be the final set of adaptations to allow animals to fully colonize dry land. And so today we see this complex interaction between plants, fungi, and animals that allows them all to utilize habitats on dry land and to thrive as colonizers of dry land. So here is a photo that shows one of those modern interactions that we see between plants and fungi. This is a fungal structure called an arbuscle or a haustorium. This is an extension of the fungi body that penetrates into plant tissue. It actually penetrates through the cell wall of plant cells, but it doesn't penetrate through the membrane of the plant cell. Instead, it comes into contact with the membrane, and you can see how highly branched it is. The plant cell membrane wraps itself around all of these branches and creates a huge surface area for transfer of materials from the fungus to the plant and from the plant to the fungus. This is a mutually beneficial symbiosis that gives the plant access to the water and minerals that the fungi can mobilize, particularly potassium, and allows for the fungus to benefit from photosynthates, carbohydrates and the like that can be transferred from the plant root to the fungus. And these are some early evidence of interactions between plants and fungi, and we see them even to this day amongst some of the fungi, not a large number of fungi, but there are about 50 species of fungi that can form these sorts of relationships with the roots of plants. We'll talk more about that when we talk about the fungi. So let's take another break. And hopefully we'll be able to come back from the break. We'll see how Zoom responds this time. But here is the concept check question. The appearance of questions. The appearance of oxygen in the atmosphere likely triggered a massive extinction even among prokaryotes at the time, extinction event amongst the prokaryotes of the time. Why? It is considered that mitochondria preceded plastids, that's chloroplasts, in the evolution of eukaryotic cells. What evidence suggests this? So I will see you on the other side of the break with the answers to these questions.